Good morning, folks, and welcome to the Global Science Show. My name is Sam. For those of you who don't know me, I'm the founder and coordinator of the Global Science Show. I'm usually found at 12 p.m. doing a chat with a scientist about who they are and what they do and why they're awesome. But we're here at 10 a.m. Jeez, it's very early and I'm on coffee 3 already. We're going to be here for about 15 minutes and we'll be back on the hour over the next couple of hours to meet a new scientist and talk about who they are and what they do. So if you want to join in the conversation, you can leave us some comments. I've already seen we've got a couple coming in on YouTube. You can also do that on Periscope over on Twitter if that's where you're watching as well. But without further ado, I'm going to get our first guest in this evening. I'm in Glasgow, and this person is also based in Glasgow. So please say hello, everyone, to Alex Wilson. Hello.
Hello.
How are you doing?
I am good, thank you. I am good. It is quite early, but ready to start the day.
Yeah, thank you so much for coming to have a chat with us this morning. I'm going to start very, very quickly because we've only got 15 minutes. So we've got a lot to chat in that short space of time. Is who are you and what do you do?
So I'm Alex, and I'm a PhD student at the University of Glasgow's Centre for Virus Research, and I work in the Brennan Lab. We look at an order of viruses called bunyiviruses, and my particular research is focused on all species of these viruses which are carried by ticks. So that's really what I do, is kind of look at these really cool viruses that are carried by ticks.
I see that we've got members of your team watching. Hi, Ben.
I can see them keeping an eye on me.
Hi, Andrew. So why ticks? What got you interested in ticks as a research organism?
So I've always really been interested in kind of viruses in general, and I always found it really cool, especially the ones which are mainly carried or transmitted to animals, but then can spill over into people. And I think ticks are quite underrated in terms of their actual ability to carry diseases. So any kind of organism that can carry a disease, we call it a vector. And tick-borne viruses are really they're not focused on as much. Like, people think ticks and they think Lyme disease, which isn't untrue, but there's so much more than ticks can carry. And because they're so hardy and they're so tough, they can transmit these diseases quite far. And especially with kind of climate change and the transportation of pets, we're seeing a real rise in them, not just in kind of ticks moving to different places, but also in the diseases they carry popping up in those places. So I'm really interested because it's something which is only going to become more and more of a thing to talk about as time goes on.
So immediately I'm getting itchy just thinking about it, like thinking about ticks. Whenever we go out, our dog inevitably gets bitten by a tick. On the west coast of Scotland, it's particularly bad. Like the first time I ever got one was on the West Highland Way, and it was the most horrifying thing that I've ever had to experience. Like they're so small, and then they get so big. Like they can get really big, right?
So they can actually expand up to about 200 times their original size. Usually if you get one which has kind of gone from really, really minuscule to absolutely massive, it's a female, because they need to take on more blood to be able to lay their eggs. And males don't tend to need to take on as much. And also if they're soft, because we've got two different types of ticks in the UK. We've got hard shell and soft shell. Hard shell ticks, the females expand massively up to 200 times, but soft shells don't tend to expand as much.
That's similar with some other ectoparasites, right? I think mosquitoes do the same thing. Like the males don't bite, or they don't need as much.
I believe it's a case of the males don't bite, but it's not my vector of interest. So
no worries.
I'm not going to comment on that one.
Won't put you in too on the spot there. So what exactly are the Brennan Lab investigating? I know there'll be a lot of things going on, but what are some of the main questions you're trying to answer?
So we've kind of got two main goals as a lab. The first is to understand how these viruses really work. So we're still a bit unclear on tick-borne viruses, how they are managed to be carried by ticks, but they don't actually kill the tick. So we really want to figure out what's controlling that balance between allowing the virus to replicate but not take over the tick completely. And we also want to know kind of where it travels to within the tick, why these viruses can't be carried by other things, say mosquitoes and things like that. And then hopefully using this knowledge and using some of the experimental systems that we have in the lab, such as reverse genetics, we can weaken these viruses, and we can hopefully either stop them being transmitted by ticks, or we can use them as potential vaccine candidates. to hopefully kind of make sure that tick-borne viruses become less of a problem in the future.
So for people who don't know, and I definitely don't mean me, what do you mean by reverse genetics? What does that mean?
So with a reverse genetic system, we have a really special kind of cell line in our lab. And what we can do with the cell line is we can take the genetic information of these viruses, so essentially what their blueprints are, and we can put these blueprints in the form of these like DNA strips, circles even, into our cells, and our cells replicate those DNA circles and allow them to actually make virus. And so if we want to say, have a look at well, what does this protein do? We can edit our blueprints before we put them in these cells, and we can take out that protein. Or we can even say tag a virus protein. So we could insert a little marker for like a fluorescent protein at the end of one of our virus proteins. And then when we put all of this into our cells and we make these viruses, they'll carry that change that we've added. And we can then use the virus stocks that are made to go and have a look at, well, if we put them in this type of cell, what does it do? How does the virus grow? Things like that.
Yeah, okay. Yeah, definitely not something that I had much of a clue about before, but like love learning, like consistently learning about all of these things. I imagine though, like trying to work with like a tiny little animal that is hard to find unless you go out and like are unfortunately bitten, but you want to work with them in a lab setting. So
Yes.
does that mean you have like ticks in the lab?
We do actually, yes. We've just, in the past month or so, we've just established our own tick colonies. So we have them in these almost glass containers called desiccators. And then they live in their own little jars with sponges at the top, which I've termed the anti-escape sponge. And we need to keep them in these sealed containers because ticks really, like, they need a lot of humidity. So in a way, they're really easy to keep because you kind of just leave them alone most of the time. But also they need some warmth, they need humidity, you need to make sure that you've got them on a day-night cycle, so they've got some really cool fairy lights around their enclosures. And then we try to feed them, we're going to be trying to feed them using artificial membranes first, where we introduce them to the membrane and there's a blood meal on top, and so they can bite and take the blood through the membrane. But yeah, it's really exciting. So we're moving from kind of doing things in our cell culture, so in vitro work, and we're moving into hopefully looking at doing it in vivo and infecting these ticks and seeing where the virus kind of goes to in the tick bodies, and then looking at how these ticks could potentially go on to then transmit by getting it to the salivary glands.
Yeah, very cool. Now you mentioned like a desiccator and fairy lights and whatnot, and it made me kind of like think about this room, like drinking coffee and not enough water, definitely drying out. And fairy lights around. Maybe I'm just a giant tick. Someone might, other people might replace the T in that word to describe me. In terms of, obviously, you've got those ones that are in the lab,
Yes.
but what about kind of wild populations of ticks? That must be like harder to come by. So is there ways that we can try and find out what's going on wild populations?
So actually, yeah, there's a bunch of different ways. So at some point, the lab will actually need to go out and collect our own ticks so that we can introduce them to our lab populations to make sure we have a bit of genetic mixing. But to do this, we get these giant flags, these white flags, and we go and kind of sweep them through the fields, and ticks will cling onto the material. So if anyone in Scotland ever sees that, don't be alarmed. We're not surrendering. We're just trying to find some ticks. There's loads of ways that the public can actually get involved as well. So at the minute, throughout the month of May, the Exploring by the Seat of Your Pants are running an initiative called, I believe it is Backyard Bio. So if anyone's really interested in getting involved in that, if they find ticks out about in the wild when they're going walking, they can tweet a picture or they can put a picture on Instagram with the hashtag Backyard Bio. And additionally, there is an iNaturalist app, which people can check out, and I will put all of the details on our Twitter and things like that. But it's a really awesome app because it means you can upload photos of anything you find, including ticks, but also fauna, animals, other insects, and you can get them identified, and there's loads of discussions going on and loads of projects. And it's just a really great way of keeping the community involved, but also we can then have a look and see, well, what are people finding when they go out and about and on walks, and whereabouts are they finding it? We're also very keen for people to send us their pictures of ticks. I'll leave the email somewhere in the Twitter thread, but it's viruses.tick.cvr@glasgow.ac.uk. So if anyone finds any pictures of anyone takes any pictures of ticks, please send them to us with where. And then coming into the summer months, we've got the backyard bio blitz, which is run by St Andrews. So essentially, we just really want to get the public involved with sending us pictures of ticks if they do find them, and that means that we get to keep an eye on ecology and kind of where they're cropping up, but also people feel like they're getting involved.
And this is part of a wider public engagement project, isn't it? Like that's run out of the Brennan Lab, called "What Makes Viruses Tick," right?
Yes, yes.
Which look at that. What a segue into, like, you can go and follow the What Makes Viruses Tick project.
So natural.
Almost like we planned it, right? For people to go and find out what's going on with this project. So Viruses Tick CVR on Twitter. And the previous people that took part in the Global Science Show today was Ben, who's part of the conversation today, tweeting about what's happening over at the lab. So yeah, what sorts of ways, like, can people? You've said like photographs and like telling us if you've seen a tick or if you found a tick, but we don't really want people to like go out looking for ticks proactively, right?
Yes, no, please don't do that. Very much.
Yeah.
Do not do that. Well, if you want to keep up with what the lab's doing, we have, of course, the Twitter and webpage, but also you can download the Spark magazine from the Glasgow Science Centre in, I believe it's through the summer months, and also you can come see us at the Glasgow Science Festival. We'll have a stall there. And additionally, we've got the Twitters, but I have just started my own TikTok. I feel very old, but I make videos about what we do in the lab and kind of the ticks and things like that. So it's just a nice kind of glimpse into research life.
I mean, it's one of those things that when we start talking about public engagement with ticks, you can't not go down the route of TikTok. Like,
yes, I was bullied into it for the pun potential.
Bullied into it. Like, there you have it. Like that would be the public engagement coordinator, Faye Watson, who was on like the Global Science Show, like going live with the Global Science Show a couple of weeks ago, bullying people into getting on TikTok. Not great. But again, those fairy lights have turned up in the TikTok as well. I've seen that, which is great. Right, we're coming towards the end of the 15 minutes. Thank you so much for coming to have a chat with us. No
worries. Thank you so much for having me on.
And hopefully folks can go and find out at the links at the bottom for Viruses Tick CVR on Twitter, or go and check out on there for other web pages and events coming up and ways you can get involved, especially with the Exploring by Seat of Your Pants Backyard Bio, which is going on throughout May, and they've got a special weekend going on today, tomorrow, Sunday, or yesterday, today, tomorrow. So many dates going on in my head. But there's loads and loads of ways that you at home can get involved in some citizen science. So thank you so much, Alex.
No worries. Have a lovely day.
I am going to use the power that I have to kick you from the room. Goodbye. Oh, first of the day, folks. I'm very much getting through this coffee, which is definitely needed. We'll be back again in 45 minutes with another guest. We're going to be talking to Adam Booth, who's a geophysicist, and we'll be talking about how we can look at what's underneath our feet. And we're specifically going to be talking about meerkats and maybe even some beavers in Scotland as well. So see you in 45 minutes, folks. And remember to check out all of the other posts that are going out as part of the Global Science Show today. See you soon. Take care. And yeah, see you in 45.